Hello everyone. We will continue lecturing on randomized controlled trials, and we will also discuss some additional issues that can arise from such studies, which is the topic of Chapter 8 from your text. Notice that I will only cover what I want you to know from those two chapters. Everything else you can skip. Here's our learning objectives for this week. We will explain why sample size is important, type 2 error, what generalizability is, how it relates to the outcome of your study. We will talk about the 4 phases of randomized trials as defined by the US government and the goals of each phase, from phase 1, 2, 3, all the way to post-marketing study. And we will conclude our lecture with ethics in randomized trials. You probably remember from your statistics class that sample size does matter. Selecting the right sample that is representative of your population of interest is also important, but even if you do select a representative sample, if that sample is not large enough, most likely you will run into a type 2 error. Type 2 error is when you run an experiment or an intervention and you conclude that there's no differences between your control and your experimental group, when in reality it is. That means you fail to reject a false null hypothesis. Type 2 errors are usually a product of too small sample size. As we increase our sample size, we gain power, and the probability of committing a type 2 or beta error decreases. Ideally, we want to control or minimize both type 1 and type 2 errors. We can control type 1 by adjusting the level of risk we are willing to take. Remember significance levels? How you set your alpha? However, type 2 errors are not directly controlled and are related to such factors as sample size. So here is a visual of all possible outcomes when we conduct a randomized trial. So we start from the top left. There is a probability that we run the experiment and you conclude that there's no differences between the two groups. And indeed, in reality, there is no difference between group A and B. Thus, we arrived in the right conclusion. Scenario number 2, there is also the probability that we run the experiment. We didn't have a large enough sample size, and although we concluded that there's no differences between the two groups, in reality, there is a difference. We just didn't have a big enough sample to get a significant result, so we end up with getting a false negative result, which is a type 2 error. Another scenario, as you can see on your second row from the left, possible conclusion is that we end up finding significant differences between the two groups, but in reality there's no differences. That means we're committing a type 1 error or alpha error. This is a false positive result, and it's also known as the level of significance. The fourth scenario is that we conclude that there are differences between the two groups and that it is actually true. That means you came to the right conclusion and you rejected a false null hypothesis. This is the correct decision, again, and a reason to celebrate since you found that your treatment works better than what is currently being used or the placebo. Hooray! Another major concern when we conduct a study is whether our findings are generalizable, meaning, can I generalize my findings to my overall population of interest? We cannot enroll everyone in a study. It is simply not feasible. Thus, the people we select to be our participants need to be representative of the population we are trying to help if our intervention or treatment works. We want to believe that the people who choose to enroll in our study are volunteers and do not have any motive to enroll in our research. Biases can arise from selecting the wrong participants. At the same time, realistically speaking, not the whole lot of people are willing to volunteer for such studies. It may be convenient to enroll people who are close by or from a hospital that you have access to, but that will not work for randomized trials. For our findings to be generalizable, we say that our study needs to have external validity. External validity is very different from having internal validity. A randomized trial is internally valid if the randomization has been properly done and the study is free of other biases. External validity, on the other hand, is our ability to apply our findings or results to a broader population. To achieve external validity, we have to think as to who is our population of interest and where can we obtain the most representative sample possible, and how to match the controls so they only differ in the exposure or disease status so we can compare the two. Of course, once we find our sample, we also randomly select them to further control for selection bias. We will talk more about the different type of bias that can arise when we design such studies. And now we're going to talk about randomized trial phases. Pharmaceutical companies always try to develop new drugs. Besides being a very profitable business, the original goal was to help people with various conditions. The U.S. Food and Drug Administration follows a standard sequence of 4 phases for testing and evaluating drugs before they're being released. Phase 1 trials involve a small sample size of patients, usually around 20 to no more than 80 people. During phase 1, toxic and pharmacologic effects are examined, which includes safety or safe ranges for human dosage and recordings of the side effects. If the drug seems to offer benefits and it is not too toxic, it will move to phase 2 studies. Phase 2 are clinical investigations with a much larger sample size of patients. Usually the number ranges between 100 to 200 patients. The goal is to test efficacy, meaning how well the drug does what it's supposed to do, and the relative safety, considering that the greater number of patients are enrolled, thus the probability of experiencing, any side effects on different individuals should be higher if the drug is too toxic. Once a drug passes phase 2, it moves forward to phase 3 testing. Since the relative safety has been evaluated through phase 1 and 2, researchers now can enroll an even greater number of patients. Phase 3 involves trials of more than 1,000 people. Recruitment can be very difficult for such large sample sizes, and ahead-of-time planning may involve recruiting patients from more than one study center. This is also known as a multi-center trial. Once again, the goal is to confirm one more time the efficacy of the drug using a greater sample of people and monitor for any side effects. During the different phases of testing new drugs, researchers may be able to observe acute, immediate side effects. Unfortunately, a few severe side effects do not show for years after treatment, such as carcinogenesis, which is the formation of cancer, or teratogenesis, that is, congenital malformations. Such extreme side effects may be infrequent, but are possible. For this reason, phase 4 studies or post-marketing surveillance are important. As more people use the drug, companies continue to monitor for side effects. Phase 4 trials are not randomized and are not exactly trials like phase 1 through 3. In order for phase 4 surveillance to be valid, it will require a high-quality system for reporting adverse effects. Of course, that is very difficult and prone to human error. A benefit of phase 4 is also providing evidence that a certain agent is indeed beneficial to many people. This rigorous testing of drugs has protected the American public against many hazardous agents. In recent years, there is a pressure to expedite the process of releasing new agents for treating such illnesses like HIV/AIDS, where there is a need for new and better drugs. But that will come with a price of possibly releasing drugs that may help treat symptoms or cure a disease, but have adverse lethal side effects. If you're interested in finding RTCs close by, here's a site that will list all major trials for various conditions. Ethics are the moral principles that govern a person's behavior or the conducting of any activity. When it comes to randomized control trials, adhering to a strong ethical behavior is crucial. People who are sick are desperate for treatment. Exploiting such vulnerable population is very easy but highly unethical. When we enroll people in our studies, we want to adhere to the most strict rules to prevent biases. But if adhering to those rules means that we endanger someone's life, then it is no longer ethical to continue with our trials. We usually randomize people to either a drug or a placebo. The drug has to offer a benefit to the participant and not put them at risk. Likewise, people who are randomized to take a placebo but require medicine to maintain their health, it is not ethical to withhold treatment in order for us to check the efficacy of a new drug. The only time that researchers can safely randomize people to a placebo is when they do not know if the new drug truly is better than the placebo. Do we ever stop controlled trials? Well, if the side effects are adverse, we should stop them immediately. Also, if we have enough evidence that a new agent is beneficial, then we should stop too. Usually there is an external data monitoring board that will make those decisions. In the US, before we conduct any type of trial, researchers need to adhere to strict ethical rules. Unfortunately, large pharmaceutical companies will try to go to other countries that their rules for conducting trials are more lax than in the US, and in some cases put people at harm's way. The first link is about the clinical trial in India, funded by multiple private and government sources, including the Bill and Melinda Gates Foundation and the National Institute of Health. It enrolled approximately 6,800 infants between March 11th of 2011 and November 5th of 2012 in rural and urban areas of the country. The experimental vaccine was given by injection in 3 doses at 6 to 7 weeks, 10 weeks or older, and 14 weeks to older. 2/3 of the infants received the vaccine and 1/3 received saltwater placebo injections. The trial showed that, as with the 2 available oral rotavirus vaccines, the new vaccine was effective in preventing severe rotavirus-induced gastroenteritis and hospitalizations. Given the availability of 2 highly effective rotavirus vaccines at the time, the India study was initiated. The failure to provide infants with one of these 2 vaccines instead of a placebo violated international ethical standards for conducting human research. The second link is about the famous Tuskegee study. In 1932, the Tuskegee Institute began a study to record the natural history of syphilis in hopes of justifying treatment programs for blacks. The study initially involved 600 black men, 399 with syphilis, 201 who did not have the disease. The study was conducted without the benefit of patients' informed consent. Researchers told the men they were being treated for bad blood, a term used to describe syphilis, anemia, and fatigue. In reality, they did not receive the proper treatment needed to cure their illness. Originally, the study was projected to last 6 months and ended up lasting for over 40 years. To make matters worse, when penicillin became the drug of choice for syphilis in 1947, researchers did not offer it to the subjects. A class action lawsuit was filed in 1973, which resulted in a $10 million settlement. Tamoxifen is a medication used for the treatment of breast cancer. It was observed that women treated with tamoxifen had a lower incidence of cancer in the other breast. Thus, a phase 3 controlled trial was initiated in 1992 to test whether tamoxifen can be used as a prevention medication for breast cancer. A total of 13,388 women were enrolled in the study above the age of 35. Women were randomly assigned to take tamoxifen or placebo. 6 years later, in 1998, an independent data monitoring committee decided that there was enough evidence that tamoxifen was beneficial in preventing breast cancer, therefore the trials can end. The same committee also noticed an increase in endometrial cancers among the women treated with tamoxifen. Apparently, the risks need to be weighted against the benefits to decide whether a medication is worth taking. Interestingly, around the same time, 2 smaller studies took place in Europe, also testing tamoxifen, but did not find the same benefit as the US study. Meanwhile, the longer tamoxifen is being used, the higher the chance of developing endometrial cancer. Hence, it is only prescribed for up to 5 years in the US. Here you can see the cumulative rates of invasive and non-invasive breast cancer occurring in participants receiving placebo or tamoxifen. In both types of cancer, women treated with tamoxifen had a decreased incidence of breast cancer. In this graph, we can also see that women treated with tamoxifen had a higher incidence of endometrial cancer in comparison with the placebo group. And here's a little sample of what some of the findings were regarding tamoxifen. It says that the meta-analysis also confirmed that tamoxifen increases risk of uterine cancer and of blood clots of the lungs. With a mean follow-up of 10 years among women with ER-positive breast cancer, 9 women who received tamoxifen died of uterine cancer, compared with 1 woman who did not receive tamoxifen. You can read the rest for yourself, but as you can see, this is pretty sad. The bottom line is that thorough investigation of the benefits and side effects need to take place before medicines are being released. Just like tamoxifen, there are other drugs out there that they can help treat one condition, but they may cause another. The question is whether the other condition is something worth risking or not. This is it for today. High five, everybody. Have a good one.